for coming. I know that the spring break is around the corner and some of you may not feel like learning about the mitochondria protein import. What does it even mean? So last time we talked a lot about the mitochondria structure and function, and today we're going to talk about protein import. And so the whole goal for this lecture is to show you that mitochondria are not completely independent organelles, and they are heavily dependent on the nucleus and on the import of a lot of protein that are nuclear encoded. They are translated on the cytoplasmic ribosomes, and they need to be successfully imported into the mitochondria in order to support and maintain correct structure and function of those organelles. And I wanted to show you this diagram at the beginning of the lecture and kind of highlight this thick orange arrow, which shows, and it goes in one direction from the nucleus into the mitochondria, and this shows that as many as 1,000 different proteins, nuclear encoded proteins, have to be successfully imported into this organelle in order to build this organelle, maintain its proper structure, function, and ensure that it works properly in our cells. So when this process is not efficient or is not working well, you can have a variety of different diseases because cells are not able to maintain healthy mitochondria and they may lose energy. So of course, as I promised, set of learning objectives. So for this topic, I refer you to chapter 12 and 14. If you feel like you need to learn more, have more information on this particular topic. So we're going to talk about proteins that are imported into the mitochondria. So we'll distinguish different categories of proteins. We'll talk about the import into the 4 different mitochondrial compartments. So there will be different mechanisms and different strategies depending where the protein will be targeted within the mitochondria. Also, we'll talk about a very important signaling sequence that are necessary for proper targeting of those proteins into the mitochondria, and also highlight the role of ATP energy in mitochondrial protein import. And I also want to talk about a special group of proteins called chaperones that play a very important role in making sure that the import of those proteins is as smooth and efficient as possible. So I just want to briefly summarize all about what we've talked last time. So we discussed that mitochondria were derived from bacteria in the process of endosymbiosis, and over millions of years they became really dependent on its host. And a lot of those genes that were originally on the mitochondrial genome were transferred to the nuclear genome. And they are factories producing cellular energy, therefore we care about those mitochondria. We want to study them and learn more and more because they are very important in our health. And we also talk about that they were crucial in evolution of complex eukaryotes. Yes, they have their own genome, and they also have a very dynamic intracellular behavior. And of course, dysfunction in mitochondria is associated with a variety of human diseases, including cancer. I'm not sure if I've mentioned this word, semi-autonomous. So mitochondria are semi-autonomous organelles, again, meaning that they rely on the nuclear protein import. So the proteins that are nuclear encoded have to be imported into the mitochondria in order to maintain their structure and function. So they are not autonomous. Autonomous would mean that they can reproduce and they can build their own structure, so they would not rely on any other organelles. In this case, they heavily rely on the nucleus. So I've already said that a couple of times, but this is very important, and I really want you to kind of, when you leave this lecture, think about this, that the effective protein import is essential for proper structure and function of mitochondria. That's the whole goal of this process. And here's a very general overview how this process works, but we'll of course talk about all of those steps in details in the following slides. So we've talked about the mitochondrial genome, and it's very small. It doesn't encode very many proteins. Does anybody remember how many proteins are encoded in the human mitochondrial genome? Very small number. I've heard the right number somewhere. Can you please? 13, yes, that's the number. That's very small. So mitochondria cannot really even support its normal function by those 13 proteins. But some of those proteins are listed here. So those are the proteins that are synthesized on the mitochondrial ribosomes and are encoded by the mitochondrial DNA. And here we have a NADH, dehydrogenase subunits, cytochrome B, and subunits of ATPase. And like I said, most of the proteins are building the mitochondrium are encoded by the nuclear DNA, and they are synthesized on the cytoplasmic ribosomes. And here we have a couple of examples. And I showed this diagram last time. Maybe we didn't discuss it well, but at this point, I want you to study this diagram. And so this is the list of major protein categories that are necessary and critically important, and they have to be, so those are all nuclear encoded proteins, and they have to be imported into the mitochondrium. So first here we have a mitochondrial RNA polymerase. So we know that mitochondria have their own genome, but in order for this genome to express the genes, or tRNAs, small RNA, they need a RNA polymerase. And they do not encode their own RNA polymerase, so they have to import this protein from the cytoplasm. They also need a DNA polymerase in order to replicate this circular genome. And again, they don't encode for this protein. So this is a nuclear encoded protein that has to be imported into the mitochondria for the mitochondrial DNA replication. And a lot of other proteins, mitochondrial ribosomal proteins. So mitochondrium encodes for tRNAs, but it doesn't encode for mitochondrial ribosomal proteins. So those proteins, those green mitochondrial proteins here shown here, they have to be synthesized in the cytoplasm and imported into the organelle to ensure that there is a translation of proteins happening in the matrix of the organelle. Also enzymes of the citric acid cycle and some other proteins as well. And I've talked about the life cycle of the mitochondria, and I want to just highlight one more time that the protein import is a critical part of the growth and division. So growth of division, in other words, biogenesis of pre-existing mitochondria. So this is, the protein import is very active process, and it happens all the time. So it's to support proper function of those organelles. And when we talk about protein import into organelles, there are two major strategies for the protein import shown here in panel A and panel B. One is called translational translocation, another one is post-translational translocation. So just by looking those two panels, what's the major difference between those two strategies? Yes, so in green we have a ribosome, and this blue thing is the messenger RNA, and this green, it's the protein coming out from this complex, and this is the signaling sequence. So what's the major difference? So in here, what's happening here? So this part, this gray part, is the membrane of some organelle. So what do we have here? What's happening here, which is different from this panel? Yes, so and this name is kind of like self-explanatory, called translational. So here we have a protein that is being actively translated, and it's in the ribosomal complex, and this whole thing is targeted to the organelle. So protein is translocated during the translation, so it's not released from the ribosome. And in this process, protein is completely translated, so the translation is finished, it's released from the ribosome and from the messenger RNA. So here we have a protein, and then this protein is targeted to the specific organelle. And this is called post-translational because it happens after the translation of the protein. And we have some examples. So this strategy is typical when you import proteins into the endoplasmic reticulum. And this strategy is specific for ER, nuclei, mitochondria, chloroplasts, and peroxisome. So of course today we're gonna, we have a question. That's the example. Maybe there are some other circumstances, but this is like a key or like a classic example when we talk about those two different strategies. Yes, in biology, I wouldn't necessarily use like yes or no, because as we learn more about those processes, we find out that under certain circumstances, you can have a mixture of different things going on. So I'm very cautious to use that this is like only, because we haven't really like explored those mechanisms completely, and there are different circumstances, different conditions that may affect those processes. So as we learn, and I'm learning, I'm always very cautious. But yes, those are the examples that we know. And when we talk about mitochondria, I would say most of the proteins which are imported into the mitochondria, this happens through a post-translational translocation. And of course, when we talk about import, what's the critical thing that protein have to have in order for this protein to be targeted to specific subcellular location? What it has? Yes, it has to have a specific signal or sequence that kind of so this sequence is recognized by other proteins, and this sequence is critically important. We call it necessary and sufficient for the import and correct localization of the protein. So in case of mitochondria, we have two different classes of sequences, cleavable and non-cleavable, as shown here on this figure. So shown here in orange are cleavable mitochondrial sequences, and those are usually very short sequences, stretch of amino acids. Typically, for the cleavable sequences, they are located on the N-terminus portion of the protein, like here, and this black arrow means that they are cleaved. So after protein goes to the right location, this signal sequence will be cleaved and removed because it's not important for the final protein. We also have a non-cleavable sequences that are positioned more into the center of the protein, and they are typically important into the targeting of the protein and telling to embed into one of the membranes, either outer or inner membrane. So they have a hydrophobic properties, and they tell other components of the import that this protein has to go to one of the membranes. Yes, but I want to again highlight that the signal sequences are both necessary and sufficient for the import. Meaning that when you don't have signal sequence, the import is not possible. So the protein won't go to the right compartment or right destination. So we've talked about the structure of mitochondria, and there are 4 possible destinations. We can have a import into the matrix, which is the most difficult to reach for the cytoplasmic proteins. Also, the second destination is the inner membrane. So those regions that we can see here. There are a lot of complexes and a lot of very important proteins that have to be embedded into the inner membrane. So, for example, we have a electron transport chains proteins, or ATP synthase. So all of those are very important proteins, and they have to be successfully embedded into the inner membrane. Also the outer membrane compartment, the area between the inner and outer membrane, and intermembrane space. So 4 possible destinations. And those proteins that are synthesized in the cytoplasm, they rely on the signaling sequences. So there is a pre-sequence and also internal sequences. And we'll talk more about those sequences and their specific features in a few slides. So when we think about the import and when we think about the whole organelle, you might wonder how this import happens. Does it happen on the entire surface of the organelle, or perhaps does it happen at specific points, location? How much do we know about this process? So this is a cross-section of the single mitochondrion. So if you would take the organelle and cut it in half, and you would look from the top. So here's the outer membrane, inner membrane, matrix. And what you can see here, you can see points where outer membrane and inner membrane come into very close contact, as shown here with this red arrow. Okay. So we have several points, contact points, throughout the surface of the single organelle, and those are the points of active protein import. So yes, so import takes place at special locations on the surface of mitochondria. And so it has a specific machinery, and this machinery is composed of protein complexes that are in the outer membrane and in the inner membrane. So TOM is a translocase of the outer membrane. So it's a huge complex in the outer membrane. I'm sorry. It's TOM. It stands for translocase of outer membrane. In the inner membrane, we have TIM, stands for translocase of inner membrane. And this is a very simplified schematic overview. Shown here is this contact site in yellow, where those both complexes, TOM and TIM, come together and like bring those membranes together and form this contact site. So here we have a cytoplasmic protein that has this nice signal sequence telling the machinery that this protein needs to be targeted to the mitochondria. And the very first step is the direct physical interaction between the signal sequence and the receptor protein on the outer membrane of the mitochondria. And this receptor protein will bring this peptide into the contact site. And then the protein will be pulled inside through this channel. So at the contact sites, there are actually a physical channels or pores, and those proteins are able to get through from the cytoplasm to the matrix. And here I wanted to talk more about those different proteins and different complexes that are actively involved in the transport of proteins into the mitochondria. So we've already mentioned about TOM in the outer membrane. So this yellow complex shown here. So you can kind of see two different parts. This is the receptor part sticking out into the cytoplasm, and also the part that forms this pore. So this protein complex forms like a hole in the membrane, like a channel or pore. And here we have another complex that is actively involved in the protein import, shown here in green. It's called SAM, and when we'll discuss the specific mechanisms, we'll discuss more about the function of this specific complex. In the inner membrane, there is also a variety of different proteins. So if we talk about the TIM, we have the TIM here shown in orange, and it also has something like a receptor sticking out into the inner membrane space, and of course the channel crossing the membrane. And there's also complex called OXA, cytochrome oxidase, for cytochrome oxidase activity. And when we'll discuss different import strategies, we'll talk about those proteins some more. And another important category that I want to mention at this point is the chaperones. So we have a cytosolic chaperones and matrix chaperones, and both they play a very important role in making sure that the protein import is tightly regulated and it's smooth and efficient. So they are very important. So here I wanted to show you this contact site in more like a 3D conformation. So this is a portion of the outer membrane and the TIM complex. So there are some subunits, there are some proteins that are sticking out to the cytosol. And here we have this complex forming a transmembrane channel, like in three-pore channel. And here on the inner membrane, this is the TIM complex that comes in, directly interacts with the TOM. And both complexes are at this contact site, and both complexes are actively involved in the protein import. And one thing that I want to emphasize here is that the TOM complex, this thing, is required for the import of all of the nuclease-encoded mitochondrial proteins. So there is no other way for a protein to enter the mitochondrium. It has to interact with the TOM complex first. And here I want to talk a little bit about the signaling sequences. So there are different targeting and sorting signals on the mitochondrial proteins. And here in green, we have this N-terminus sequence, which is positively charged. And this is a cleavable sequence, a very short stretch of amino acids. And this sequence signal as a signal to the matrix. So it's called matrix targeting signal. And we have also a transmembrane domain signals shown here in purple. So several mitochondrial proteins need to be embedded into the membrane, so therefore they have a transmembrane domain signals. And some proteins, they also have a sorting signal shown here. It's cleavable. So the hydrophobic domain flanked by two hydrophilic sites, both sides here. And this signal is specifically, it's present in proteins that have to go to the either to the intermembrane space or the inner membrane. And in case you, so this is a very nice figure, legend for this figure taken from the paper. So now I want to talk about the pre-sequence. So pre-sequence is important. It's present in all of the proteins that need to go to the mitochondria, and it sits on this very N-terminus portion of the protein, so it's N-terminal. It's short, 15 to 55 amino acids. So here we have this nice stretch of amino acids. And it's very unique because the color coding reflects chemical properties of those amino acids, and when this stretch of amino acids is making the secondary structures, making the alpha helix, it's gonna put all of the charged amino acid into one side of the helix, and all of the hydrophobic amino acids will be clustered on the opposite side. So you will have a helix, very short helix, with two faces or two sides. One will be hydrophobic and another one will be charged, positively charged. And therefore it's named amphiphilic alpha helix. And it's very important sequence because this is the first step when the protein will make a physical interactions with the receptors on the surface of the mitochondrion. So this figure shows a little bit more details about this important step. It's just showing that so those hydrophobic amino acids are making direct interactions with the TOM20, and then the helix is positioned in the way that those charged amino acids will interact with the TOM22. So TOM20 is this receptor portion of the TOM complex, and TOM22 is the channel portion of the TOM complex. So and yeah, we also said that TOM is required for the import of all nucleus encoded mitochondrial proteins. So now we'll talk about import of the proteins to different destinations, and we'll start from the most distant destination, the matrix. And before that, I just wanted to talk a little bit about what's happening on the cytoplasmic side. So after translation, so let's think about those 1,000 nuclear encoded proteins. So after translation, they are sitting in the cytoplasm and just waiting. So what's happening after translation? So they are released from the ribosome. They are released from the cytosolic ribosomes, and then they interact with the cytosolic chaperones like HSP70, and this is the key step, the interaction with those chaperones, because chaperones are important in order for those proteins to keep an unfolded state. They also prevent aggregation of those proteins, and they also prevent degradation of the mitochondrial targeted protein by the cytosolic proteases. So I want you to remember the key role of cytoplasmic chaperones. So they bind those proteins and they protect them and prevent from aggregation and degradation in the cytoplasm. So now we're going to go over the mechanism, and there will be 7 major steps. So here we are targeting the protein into the mitochondrial matrix. There will be 7 major steps, shown here as those black squares. And I will break down this whole mechanism into 3 different parts. So first we'll focus on this part. So this is the cytoplasmic portion and the outer membrane. Then we'll talk about this transition through the contact site, and then the final steps, what's happening in the matrix. So we kind of like talk about that already. So here we have a protein that has the matrix targeting sequence, this amphiphilic alpha helix shown here in purple, and the rest of the protein. So this is called precursor protein, and it's bound by those chaperones shown here in orange, and they use ATP energy. So just please remember that those chaperones use ATP energy in order to maintain this precursor protein in the unfolded conformation and in order to protect this protein from aggregation and degradation. So ATP energy here is very important. And then the second step, what's happening here. So in the second step, there is a direct physical interaction between this signaling sequence and the receptor portion of the TOM complex. So this green structure is the receptor, TOM 2022. So there is a direct physical interaction between those, between the peptide and the receptor. Now let's focus on this part. Step 3, 4, and 5. So then this peptide is transferred from the receptor to the channel portion of the whole complex, of the whole TOM complex. And what we have here, so this signaling sequence go first, is going through this channel, and then so this is the contact site where 2 complexes, TOM and TEAM, are kind of aligned in this conformation. So they form this like a very long straight channel, and the protein is going through this channel, passing through the intermembrane space and going straight into the matrix here. And also in matrix we have a chaperones, a different category of chaperones. They are called matrix chaperones. So they are different from cytosolic chaperones. So they sit in matrix, and here at the bottom of this TEAM complex, they use ATP energy and act like a motor to pull this peptide inside. Using that ATP energy, they are pulling this protein inside the matrix. And when the whole protein is translocated through this contact site into the matrix, we have a cleaving of this matrix targeting sequence. So there are matrix proteases that are responsible for cleaving the signaling sequence. So the signal sequence needs to be cleaved in order for this protein to acquire native final conformation. And final steps, so after this signal is cleaved, we have so the protein that was targeted to the matrix can acquire the native conformation, and even it's not shown here, but there is a separate group of chaperones that help this protein here to to assume its native conformation. And I really wanted to talk a little bit about those chaperones that are present into the matrix. You have a question? Yes, so in this case we talk about this very N-terminus cleavable sequence. So this sequence gets removed always, all the time from every protein. It needs to be degraded.
Okay, so for those proteins that have uncleavable sequence, are they not?
We'll talk about that in a few slides. So those proteins that have uncleavable sequences are typically embedded into the membranes. And those uncleavable sequences are important domains, hydrophobic domains, to anchor the membrane proteins. And we'll talk about that. So I just wanted to talk a little about the chaperones and their amazing structure. So they look like those barrels, and the barrels are formed by those 7 subunit rings-like structures. And they are able to use ATP energy, and they are able to interact with the unfolded proteins. So they interact, they use ATP energy and kind of like unfold the peptide, refold, and do it several times using ATP energy. And finally, when the protein acquires its native structure, the final native conformation, which is usually energetically favorable state, then the final protein is released. And here we have a really nice figure showing this whole process. So this is the barrel of the chaperone, and here we have a unfolded protein. So it's gonna enter into the cavity of this chaperone here, and then this chaperone also interacts with a different protein. This is a HSP10. It's like a helper chaperone. So this peptide will enter inside, and this helper chaperone will cover the whole barrel. So here we have like the capsule, and there is a unfolded protein inside. So this whole structure will use ATP energy, do its magic, unfold and refold this peptide several times, and then finally will release the fully folded protein. And then it will disassembly and can do the cycle all over again. So this is a very, very interesting system. And of course, role of ATP energy in the mitochondrial import is very important. There are several steps throughout the process that use and rely on ATP energy. So we've talked about cytoplasmic chaperones, we've talked about the matrix chaperones. Another important point is here is the membrane potential. So the matrix is negatively charged and the intermembrane space is positively charged. And you may remember when I was talking about those signaling sequences, the alpha helix is positively charged. It has a phase that is positively charged. So it will be attracted by the negatively charged matrix. So those electrostatic interactions also play an important role. And I talk about the chaperones that are working like a motor proteins, like pulling the peptide inside, and also chaperones that help with the final folding. So all of those steps, I just want to highlight the importance of the ATP energy and the importance of those different complexes relying on the ATP energy. So now we're gonna move on and talk about the import into the inner mitochondrial membrane. And there are three strategies, three different paths, and we'll discuss separately each of the strategy. And so different strategies are suitable for different proteins, depending on what type of protein it is. And so this is the time when we'll talk about those non-cleavable sequences and their function. So here we have a path A. So this is the precursor protein, and it has the matrix targeting sequence shown here in pink, and the red is the stop transfer sequence. So of course we are very familiar with those initial steps. Every protein has to go through the Tom complex, so we've kind of covered these interactions with the Tom complex. So what's happening here? So this entire peptide is translocated, but not completely into the matrix. Just this pink targeting sequence will be translocated into the matrix, and it will be cleaved. And then what you see here, so here is the stop transfer sequence, the hydrophobic sequence that is a signal for other proteins to embed this peptide, to embed this protein into the inner membrane. So here is the final protein embedded into the membrane. So this is this non-cleavable sequence, which is now part of the transmembrane domain. So now we're going to talk about a different scenario, path B, and what's different here is that we have a special sequence. So there's one more sequence, the hydrophobic segment, and it's called OXA1 targeting sequence. So what's happening in this scenario? In this scenario, entire peptide will be first imported into the matrix. So the whole thing will be imported. This pink signaling sequence is cleaved, as you can see here. So we end up with the kind of unstructured, unfolded protein that has two non-cleavable sequences present. And then this protein will be processed by this inner membrane complex called OXA. We talked about OXA when we were talking about inner membrane complexes. So OXA will process this protein and embed the protein into the inner membrane. So it's a slightly different strategy. The path B is slightly different strategy as compared to path A. And we also have path C. What's different here? We have like several multiple stop transfer sequences, so those non-cleavable internal sequences, and all of those sequences will be processed by a different complex, TIM22, TIM54, in the inner membrane, and so this entire peptide will be embedded into the inner membrane. So now we'll talk about import into the intermembrane space and finally outer membrane. So intermembrane space, what do we have here? So the strategy here is that for the protein to go to the intermembrane space, it has to have this intermembrane space targeting sequence. So just make sure you remember that. This is shown here in purple, and it's gonna go through the outer membrane first. It will go partially through the inner membrane, so this targeting, this pink sequence, this N-terminus sequence will be removed, and then this protein will be processed now by protease. So protease will degrade this targeting sequence portion, and the protein will be released here. Here's the intermembrane space, will be released and will acquire, it's final confirmation. So here's another example, but I don't think we have to really talk too much about that. It's very simple and straightforward. And finally, a very easy way, the most accessible compartment is the outer membrane. And so this figure outlines the process. It's again very simple. So the protein goes into the intermembrane space. Chaperones are helping here. And the SAM complex in the outer membrane will make sure that this protein will acquire its final native conformation and it will be embedded into the outer membrane. I have 2 more slides and then we'll have a short quiz. So this is an assay that I wanted to tell you, and it will be very useful information for the next breakout session. So all of this information about the sequences, cleavable, non-cleavable, scientists were able to obtain this information by using and performing this assay. It's called trypsin protection assay. And what you can see here, so this is the outline of this assay. So here we have our mitochondrial protein, which can be expressed, for example, in yeast cells. So it can be expressed and purified. So here we have the protein and the uptake targeting sequence shown here in red. And we can also purify entire mitochondria from cells. So in this experiment, what we are doing, we are taking the purified proteins and we are taking the purified mitochondria and mixing both together in this test tube and incubating for some time and then adding trypsin. Does anybody know what trypsin does? What is trypsin? Yes, it's an enzyme which degrades protein. So what's happening in this test tube? So we have the peptide and the mitochondria, and it has the signaling sequence. So if everything goes well, our peptide or our protein will be imported somewhere into the mitochondrium, somewhere. It can go to the matrix or it can go to one of the membranes. But it will enter inside if the import is successful. And so what's going to happen when you add trypsin, all of the protein that could not be imported successfully will be degraded, and only the protein that was imported successfully will be protected. So here when you add trypsin to this tube, this trypsin shown here as green, it's going to degrade all of the protein. And then you can kind of like work with the mitochondria and isolate or purify your protein of interest, and then you can even test where it went. So this assay was very helpful in kind of like studying all of those sequences, what is necessary for the protein to be targeted into the mitochondria, how much of the sequence, do we need 5 amino acids, do we need 20, so this was a very important assay. And the final slide, so this is the outline taken from the textbook, so if you need to read more about that, please go to the textbook. But so this is the approach where we can isolate entire organelle from the cells, and we can also, by doing a centrifugation and also switching from different osmotic media, we can separate different compartments. So we can separate the matrix content, we can separate the outer membrane content, inner membrane. So this is again very useful, very nice tool. And I think I'm gonna stop here. So we'll have short quiz. So I already talked about this stuff, so it's very straightforward. Do you need my help in? No. Sure.
I think one might be enough. I think we have less than 100 students.
Yes.
It's hard, right?
Yes.
Five minutes, 10 minutes, I don't know. Is it complicated?
No, it's not complicated, but I can take as much time as I can. So after you finish your quiz, you're free to go. And I wish everybody a happy spring break.